Hello, can somebody please confirm you can hear me?
I can hear you.
Thank you. All right, before we begin, are there any questions? For some reason, my computer's kind of slow today. Let me share my screen here. Ah, there, you should be seeing my screen. Come on. There we go. All right, we were on this slide. Let me show the syllabus. What's going on there? I don't know what happened to the Zoom. I didn't find an email about it, and I'm not sure what's going on. And I need to ask, I guess, the Zoom people what they did because, or maybe it was the Canvas people. One of them did something, and I don't have access anymore to the original meeting or my meeting things. And I noticed it also changed. I don't easily have access to um, the older meetings. I can access an older meeting, but I can't open it. And anyways, I had to find another way to access the class. So that's why I'm a little late here. All right, today is February 24th, and we're supposed to be finishing chapter 8. I don't think we'll get there, but let's give it a try. Um, there is no lab today. I will be opening the lab for about 15 minutes. After the last 15 minutes, or the last student question, I will then log off. So you only need to show up at the lab if you have a question. If you don't have any questions, you do not need to show up at the lab. Any question about any of that? All right, let's get to chapter 8. So we were talking about the complications of RNA transcription, and we mentioned with prokaryotes, we have the complication that besides multiple messenger RNAs can be made at the same time on the same gene, you can have multiple um, ribosomes reading the messenger RNA, making multiple proteins on that messenger RNA, even before the messenger RNA is finished. Is that clear to everyone? So in prokaryotes, because this happens in the cytoplasm, you can have translation beginning before transcription ends. In a eukaryote cell, you don't have that problem because transcription happens in the nucleus and then translation happens in the cytoplasm. But we have another complication, and that is in eukaryotes, for a gene, we have regions of the gene called exons, which code for the finished gene product, which if it's a protein, it codes for the protein, but it could be a messenger RNA, remember that. And those we call exons that actually code for the gene product. We also have regions inside the gene called an intron that do not code for the finished gene product, meaning they do not code for amino acids if we're making a protein. These introns have to be spliced out, that means removed, and then the exons spliced together, combined. So we're using splice to be cut out and then stick the exons back together. Any question about any of that? So initially in a eukaryote, you get an RNA transcription, which makes transcript of both the exons and the introns. The introns have to be spliced out, and then the exons spliced together. And only the finished messenger RNA, which has the introns removed, can leave through the pores of the nucleus. If it has an intron in it, this RNA transcript cannot leave through the pores of the nuclear membrane. And then that finished messenger RNA goes into the cytoplasm and it can be translated into protein. Any questions about RNA processing in eukaryotes? So an intron is another part of DNA that does not code for the functional gene product. If you remember, eukaryotes have a lot of that DNA. Most of it is in between the genes. So we have one gene here and then one gene here, and there's non-coding DNA between it, but there's also non-coding DNA inside the gene, and that we call an intron. Any question about any of that? All right, so we've now talked about the structure and function of genetic material. Let's move on to the regulation of bacterial gene expressions. We'll only talk very briefly about gene expression in a eukaryote cell, but it's similar, and they both use operons to control gene expression. Now let's talk about that, and then we'll move on to mutation, talk about genetic transfer and recombination, and lastly, we'll talk about genes and evolution. Don't know if we'll get to all of that today, but let's talk about the regulation of bacterial gene expression. One way to regulate a cell is to control the expression of the genes. We have some genes which are expressed at a fixed rate and the gene is always turned on, such as a constitutive enzyme or a constitutive gene, where the gene is always on and it's always expressed at a fixed rate. There are other enzymes that are expressed only as they are needed. And you can have a repressible gene or an inducible gene having a repressible or an inducible enzyme. Any question about any of that? So these genes are only transcribed when they are needed. And this is regulation by a pre-transcriptional control, meaning we're regulating the expression of the gene before transcription, pre-transcriptional. We'll talk about inducible operons, and we'll talk specifically about the lac operon, which is an inducible operon, and the only operon we're going to talk about. But we do have repressible operons. You're not tested on it. We're not going to cover it. They're similar to an inducible operon. The difference is an inducible operon is a gene that is normally turned off, but when it is needed, it is turned on. A repressible operon is one that is normally turned on, but when it's not needed, it's turned off. And like I said, we won't cover it and you're not going to be tested on it. There are also epigenetic control means of controlling a gene, and you should be lucky we're not going to talk about that. You're not going to be tested on it because epigenetic means for controlling gene expression, they've been studying it now for, gosh, I would say about 100 years, and we still do not have a good understanding of how epigenetic control means control gene expression, meaning turning on and turning off a gene. Any question about any of that? There are also post-transcriptional control means for controlling the expression of a gene, And we're not going to talk about it. You're not going to be tested on it, but this would be something like you make the gene product and you make the protein, and then you do something post-transcriptional control to control the expression of the gene, like you can inactivate the protein, or you can simply degrade the protein. Any question about any of that? All right. So we're not going to talk about post-transcriptional control, repressible operons, or epigenetic control mechanisms. We're only going to talk about inducible operons. When we're talking about a bacterial operon, you should realize that an operon is sort of like a super gene. Normally, when we're talking about a gene, let me blow this slide up. Like the human blue-eye gene or the lactase gene, the gene encoding the enzyme lactase. We're talking about a structural gene. But the operon is regions around that structural gene that can control the structural gene. And yeah, they may have more than one structural gene in an operon. Generally, structural genes are involved in a single metabolic pathway and have coordinated expression. For example, in the bacterial operon, for the lac operon, we have three structural genes in one operon. The Z gene is the gene encoding the enzyme lactase. It makes the enzyme which can break down the sugar lactose into beta galactose and glucose. I have to remember my sugars there. Also in that gene is the gene Y, which I think is sometimes called permease, and it encodes the what is that called? The protein. The protein that the transport protein that brings the sugar lactose across the cell membrane. So the Y gene, when it is expressed, puts in the cell membrane the transport protein that can then bring the sugar lactose into the cell membrane. And then the A gene is a gene which helps convert the sugar galactose into the sugar glucose. So all three genes in the lac operon are involved in the metabolism of the sugar lactose. And that's always what you see in a prokaryote operon. If there's more than one structural gene in the operon, they're all related. And in the case of the lac operon, we have three genes which are all related to the metabolism of the sugar lactose. Eukaryote operons are similar, except that they only have one structural gene in each operon. But they do have more regulation controlling the structural gene. For example, eukaryotes have the gene encoding the enzyme lactase, so it's the Z gene. They also have the Y gene encoding the transport protein, bringing the sugar lactose into the cell membrane. Sorry, didn't mean to do that. And then they have the A gene converting the sugar or helping convert the sugar beta galactose into the sugar glucose. So they have all three structural genes. They just have them not in one operon, but they have them in three separate operons because each operon in a eukaryote has only one structural gene. An operon also has the controlling regions around the structural gene. In the case of the lac operon, we have the P region, the promoter, upstream of the Z gene, and we have an O region, also upstream of the Z gene. And both of these are controlling regions that control the expression of the structural genes, in this case, the Z gene. Also, a part of the operon is the terminator, which will be after the end of the structural genes. And you should know what the terminator does. I've already told you. The terminator codes for the RNA polymerase to fall off of the DNA. And so that stops transcription of the gene. And then the P region, I've already told you what that does. The P region in the operon is a controlling region coding for the promoter, which is the region where RNA polymerase binds to DNA. And the RNA polymerase will bind to DNA and then move down the DNA, transcribing the DNA into RNA after the promoter. And that will continue until the RNA polymerase reaches the terminator, and that'll stop transcription. Any question about any of that? All right. The lactose operon is an inducible operon. That means it is normally shut off, but it can be induced to be turned on, and it will be turned on when the cell, because we're talking about a bacteria, needs the genes to metabolize the sugar lactose. Any question about any of that? All right, normally the lac operon has another gene called the repressor gene, and this is not a part of the lac operon. It has its own operon, but this repressor gene makes a repressor protein which binds to the operator. And when the repressor protein is bound to the operator, when the RNA polymerase then binds to the promoter and starts moving down the DNA, it gets blocked by the repressor protein. And that shuts down the transcription of the lac operon, meaning the Z gene will not be transcribed, as well as the Y and the A genes. And that's what the repressor protein does. It's another gene on another operon that simply makes a repressor protein, and it's an active protein, and that means it binds to the operator, and then that shuts off the expression or the transcription of the lac operon. Any question about any of that? Normally, in E. coli, as well as any other bacteria, the lac operon is turned off by this means. Why do you think a cell would want to have the lac operon normally turned off? What do you know about the sugar lactose? Come on, somebody's got to know something about the sugar lactose. Where do you find the sugar lactose?
In milk?
Yeah, in milk and dairy products. How often would E. coli, if it's not in somebody's gut who is drinking milk or actively eating dairy products, like me, I don't eat much dairy products because I'm lactose intolerant. But let's say you're an E. coli on an apple on the orchard in the farm. How often would E. coli see the sugar lactose if E. coli was sitting on the apple? How often would
Probably
the
like
apple?
never.
Yeah, probably never. The milk is never, or milk dairy product isn't probably never going to get on the apple. And that would be true for E. coli in the ground or in the ocean or most other places. Obviously not in someone's gut if they are drinking milk. But and the only other time E. coli might see the sugar lactose is if E. coli is on a dairy farm. Okay, so E. coli doesn't normally see the sugar lactose. So E. coli would want to shut down the the lac operon because it doesn't need any of these proteins. And that is why the lac operon is normally turned off, why it is an inducible operon, because E. coli is not likely to see the sugar lactose. But what happens when E. coli sees the sugar lactose? We have to have a means for turning on the the lac operon, because you'd want to be able to make the proteins to metabolize the sugar lactose. What happens is whenever lactose is around, we have an isomer of lactose called allolactose, and they're the same sugar, it's just one is left-handed and one is right-handed. And if you remember what an isomer is, it's the same molecule, it's just one's left-handed, one's right-handed. Allolactose, you always have it around whenever the sugar lactose is around. And allolactose is the inducer, which binds to the repressor protein. And when allolactose binds to the repressor protein, it makes the repressor protein inactive. And now the repressor protein cannot bind to the operator. If the operator doesn't have the repressor protein on it, then when the RNA polymerase binds in the P region, the promoter, it will then move down the DNA, transcribing it, and we'll get transcription of the lac operon. Any question about any of that? Well, what do you notice which is a little different about the lac operon or the transcript of the lac operon compared to every other gene where we've talked about transcription? How many proteins do you get from this one messenger RNA? Come on, somebody's got to see it.
Is it 3?
Yeah, 1, 2, 3. We've never seen that before. But in the case of the lac operon, you will get 3 proteins from this one messenger RNA because the operon has more than one structural gene in it. And we get 3 proteins made. Beta galactosidase is another term for the enzyme lactase. Permease, already mentioned, is the transport protein that brings the sugar lactose into the cell. And you don't need to know about transacetylase, but that's the protein which helps convert the sugar galactose into the sugar glucose. All right, let's watch a little movie on the lac operon. Can't seem to open it here. There we go. Oops. I need to shut that down. Something, oh, there it goes. Told you my computer is slow for some reason today.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon: beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
All right, any question about that? The movie was a little oversimplified, like it said lactose instead of allolactose, but when lactose is around, allolactose will always be there. All right, any questions about the lac operon and the operation of the lac operon? All right, let's move on to mutation. Mutation is a change in the genetic material. Mutations may be neutral, beneficial, or harmful. A mutagen is an agent that causes a mutation. Here we're looking at a mutation. There are mutagens which can cause a mutation, but mutations can occur spontaneously, where the mutation occurs in the absence of a mutagen. There are different, let's go back here and state that a mutation can happen spontaneously just when the DNA is being replicated and the DNA polymerase inserts an incorrect base pair. That can cause a mutation. Of course, the incorrect base pair has to be replicated and then it's a mutation. Before it's replicated, the cell could always repair that DNA because it'll note that the DNA is not hydrogen, the incorrect nucleotide that's been added will not hydrogen bond with the second nucleotide in the other strand of DNA. And so the cell could always correct that mutation. But once the cell replicates, that mutation will become fixed, and then it's almost impossible to reverse that. It can be reversed if you happen to have a reverse mutation, but then you need 2 mutations in the same nucleotide. And I have to reverse it correctly to the correct nucleotide base. All right, there are different types of mutations. There's a base substitution, an AKA a point mutation. This is the most common type of a mutation, a base substitution, but you could have a frame shift mutation and a macro mutation. We'll talk about each of these in a little bit. Let's mention that the base substitution, the point mutation, could result in a silent mutation, a missense mutation, or a nonsense mutation. A frame shift mutation is where a extra nucleotide base is inserted in the DNA, or a nucleotide base is removed from the DNA, and that could be a multiple number of bases added or removed, except for 3. And the reason why it's except for 3 is if you add one extra base, you don't have your normal 3 codons, you got 4 codons, but the codons are read in groups of 3, so that will change that codon. But then the next codon will start with that 4th base, and then we'll take the next 2 bases. And so you can see that if you add or remove a base, you change all the codons after that addition or subtraction. Does anyone have a question about that, or is that not clear to anyone? If you make a frame shift mutation by adding or removing a base, you change the codons from that added or removed base all the way through the rest of the gene. And that's why they call it a frame shift mutation. You change the reading frame of the codons. A macromutation is where an entire region of a chromosome is added or deleted. Let me mention that a macromutation is not discussed in your book, so you'll only get it from me in the slides. One example of a macromutation is Down syndrome, where the entire chromosome is actually extra. I think it's chromosome 21. Can somebody confirm that? Down syndrome, is it chromosome 21 or is it 23?
I can't remember. Hold on, I'll Google it.
Oh, it's fine. Anyways, it's either chromosome 21 or 23. I think it's 21, but it could be 23. I'm supposed to have that on the slide here, so I'd remember that because I never remember 21 or 23. Anyways, those are the 3 types of a mutation. Any question about any of those 3 types of mutations? All right, let's talk about a base substitution mutation. This is where one nucleotide is substituted for another nucleotide in the DNA. That we call a base substitution. Oh, let me go back here. This was a paper I read about. Spoons were sticking to the skin of this child, and that you can see some of the spoons there. Let me blow that up. These spoons are sticking to the skin of the child, and they were checking to see if this child was actually a mutant, like Magneto, who was born, if anyone knows X-Men. I never read anything further about what the study came about that, but like I said, they were checking this child to see if he was a mutant, and spoons literally stuck to him. He might not have been physically magnetic, he might have just had very sticky skin. All right, I talked about base substitution here. Let's move on. You can have a silent mutation, and that's where you change one base in the DNA, but that base codes for the same amino acid in the codon, meaning we have UUU for the RNA, which can be mutated to UUC, but both of these codons code for the amino acid phenylalanine. This is called a silent mutation, where the DNA is changed, but the amino acid sequence in the protein is not. There is no effect on the protein because it's coding for the same amino acid, so the protein is the same. Any question about any of that? And that's called a silent or a neutral mutation. I'll call it a silent mutation because a neutral mutation could have the amino acid changed and still be called a neutral mutation, like the brown-eyed human allele for eye color was mutated about 10,000 years ago in Northern Europe to the blue-eyed allele, causing blue-eyed to be born in the human population for the first time. that was a neutral mutation. At least today it's neutral mutation, meaning the blue-eyed allele doesn't have any reproductive advantage over the brown-eyed allele, and vice versa. So it's a neutral mutation, but it's not a silent mutation because it codes for a different amino acid, and that changes the protein. And one protein makes the eye color look blue, the other protein makes the eye color look brown. Any question about any of that? So I'll call this a silent mutation because you can have a neutral mutation, which is a silent mutation because that's obviously a neutral, but you can also have a neutral mutation which changes the protein. All right. You can also have a missense mutation. That's when you change one base in the protein, or one base in the DNA, sorry, and you change one amino acid in the protein. For example, here we have oops, not that one, it's GGC, coding for the amino acid glycine. You can have the mutation, switching the G to an A, and then it's going to be the codon AGC, which doesn't code for the amino acid glycine. It codes for the amino acid serine. And this is a change in the DNA and a change in the amino acid sequence of the protein. And this we call a missense mutation. It can change the protein. An example of a missense mutation is the sickle cell anemia gene, causing sickle cell anemia, as well as the blue-eyed gene is a missense mutation from the brown-eyed gene. Any question about any of that? We can also have a nonsense mutation where you change one base in the DNA. What are we getting here? Lysosine, so switching from AAG, encoding the amino acid lysine, to the stop codon. You don't need to know the sequence, but UAG, coding for stop, and that reduces the size of the protein. Here we only have one amino acid. Normally the protein is truncated because instead of being to here and then the stop will be in an earlier location, and so you'll get a truncated protein. Oftentimes this will totally inactivate the protein because a stop makes a smaller protein. You need the entire protein to have the protein functional. Anyways, that's a nonsense mutation where you change the base, which changes the amino acid to the stop codon. Any question about any of that? Oops. All right, let's talk about the frame shift mutation. So here we have the amino acid methionine, lysine, and then phenylalanine, UUU, but we subtract an amino acid, pulling out the A here, and then the amino acid, instead of being UUU, coding for phenylalanine, it'll then code for UU, and then the A is pulled out here, so the C will be corresponding to a G, and that will change this codon. Instead of UUU coding for phenylalanine, it'll be UUG coding for leucine, and the G is just pulled up from the next codon here, and that's where that G comes from. From the next codons pulled up to this codon, because codons are always read in groups of 3. And then the next codon, instead of being GGC, coding for glycine, in this case is going to code for GCU, pulling the U from the next codon. And you can see this will be changing the protein everywhere from this frame shift mutation. And frame shift mutation is just the insertion or deletion of one base, in this case, or the insertion or deletion of 2, 4, whatever, everything other than a multiple of 3. So 1 or more bases other than a multiple of 3 results in a frame shift mutation. After this change, the entire coding reading frame is changed. That will drastically change the protein, oftentimes making the protein non-functional. What happens if you remove 3 bases? So here, instead of removing just one base, AAA, we remove all 3 of these A's. Then you just have a protein without the amino acid phenylalanine, but the entire protein after this is the same protein. Meaning glycine will be there, stop codon will be there, assuming the stop codon is the next one. But the point is, this amino acid is removed, and then all the other amino acids are correct. That is a frame shift mutation.
So
It's
it
not a
can
frame
it
shift
can be
mutation. Sorry, that's not a frame shift mutation because the frame is not changed. It is a protein missing one amino acid. All right, what was your question?
oh, so it can't be more than one base for it to be a frame shift mutation.
If it's a multiple of 3, then it is not a frame shift mutation. Because if you have a multiple of 3, let's say we remove AAA, but come on, TTC as well, then we'll be missing these 2 amino acids, but that won't be the same, that won't be the same, the stop codon will be the same, so that's not a frame shift mutation. You're just missing these 2 amino acids. A frame shift mutation is where you change the codon reading frame. And then after that point, the codons are all different after that. Any question about that? So a frame shift mutation has to be removing 1 or 2 or 4 or 5 or 7 or something like that, or adding 1 or 2 or 4. But if ever you add or subtract a multiple of 3, that could be 3, 6, 9, that is not a frame shift mutation. Because you don't change the reading frame of the protein. You just remove or add extra or some amino acids. All right, let's talk about the causes of mutation. There are 2 causes of mutation, a spontaneous mutation, and this mutation can happen on its own. It's just when the DNA gets replicated, a mistake gets made. And there are also mutations caused by a mutagen, where the mutagen increases the frequency of the mutation, causing the mutation itself. There are 2 types of mutagens, radiation and chemical mutagens. In radiation, there's 2 types of radiation that can cause mutation. There's ionizing radiation. This is when X-rays or gamma rays have a lot of energy associated with the radiation. And when they hit an atom or an ion or a molecule, that energy can be absorbed by an electron. And there's so much energy that that electron will leave the atom or the ion or the molecule, making this atom minus an electron, and that we call an ion. It's going to have a +1 charge because this one electron has been left. We won't talk about how we can repair that. But that's how ionizing radiation can cause a mutation. By creating an ion, this ion can then bump into DNA, steal an electron, and then mutate the DNA. Of course, the X-ray or the gamma ray can directly hit the DNA and directly damage the DNA as well. You can also have a mutation caused by ultraviolet light. Ultraviolet light is a form of non-ionizing radiation, but you should realize that visible light, meaning what we see with, is also a form of non-ionizing radiation. Visible light does not cause mutation. Only UV radiation causes mutation of non-ionizing radiation. Let me build this up a little bit. What UV light does when it strikes DNA is it hits two thymines, which are right next to each other, and then it causes the thymines to form a dimer. That's two chemical covalent bonds between the thymines. There's no normal bonds between two thymines right next to each other. The thymine does not hydrogen bond, or even, excuse me, it doesn't, well, it doesn't hydrogen bond to the thymine next to it, but it also doesn't form a chemical covalent bond between the thymine next to it. When UV radiation hits two thymines that are next to each other, it causes two chemical covalent bonds to form between the two thymines, and that's called the thymine dimer. That causes a bubble in the DNA, kind of showing like here. This bubble occurs because these two thymines do not hydrogen bond to the A's next to them. And the DNA can actually make use of that to remove the damage and remove the thymine dimers. We're not going to talk about that, but the cell can repair that by removing the thymines and then inserting new thymines. And that's how UV radiation causes mutations in DNA. This DNA, where the bubble is, cannot replicate or cannot be transcribed, at least correctly. That bubble stops DNA replication and transcription. All right, any question about how radiation can damage DNA? If not, let's talk about how chemicals can damage DNA. I'm only going to show the example of how nitrous acid, a chemical, can act as a mutagen. When nitrous acid gets in a cell, it can bind to DNA and then change this A here so that the A is no longer recognized by the DNA polymerase as an A. And when the DNA polymerase will see this changed A, instead of putting a T here, it will read this changed A as a G and then insert a C. When this DNA then replicates, this change then becomes permanent. Instead of a T being here, which is still seen here, this will become a GC change. That will be a mutation. The only way you could go back to this is if you had an exact reverse mutation where this G were to be changed into an A. And that could happen, but it's very unlikely to happen because you need to have that nucleotide change and it has to mutate directly into an A, because if it mutates into another nucleotide, it won't reverse the mutation. You'll still have the mutation there, just be a different mutation. Any questions about how nitrous acid can act as a chemical mutagen? Just binds to the DNA, changes this A, that change J is not read as an A, it's read as a G, and then that will mutate the DNA. Any question about chemical mutagens? All right, the frequency of a mutation happens spontaneously for every 10 to the 9th, 1 times 10 to the 9th replicated basis. We get one base, which is a mistake. And so the spontaneous mutation rate is for every 1 times 10 to the 9th replicated basis, we will have one base being a spontaneous mutation. Because each gene has about 1,000 replicated bases in the gene, you can say the mutation rate is one gene and 10 to the 6 replicated genes will be a mutation, meaning this is the same number as that number. This is talking about mutation and numbers of replicated bases. This will be the mutation rate per replicated gene. A mutagen just increases the mutation rate by 10 to 1,000 times greater, where a mutagen hits the DNA, and then instead of being 1 and 10 to the 6 replicated genes being mutated, we will have between 1 times 10 to the 5th replicated genes to 1 in 1,000 replicated genes being a mutation. Does anyone know what the most common mutagen is that humans get exposed to? Anyone a smoker? Cigarette smoke is a mutagen, which is the most common mutagen other than UV light. I guess I forgot about that. That humans are exposed to. Obviously, UV light is a mutagen. I already talked about that. And the good news is our skin helps protect us against UV light. All right, identifying mutants experimentally. You can test for a mutant by a positive or direct selection or by a negative indirect selection. A positive direct selection detects mutant cells because they grow or appear differently. For example, if this individual were to go walking down the street, people would say, yep, there goes a walking mutant. Because the person looks different. A negative or indirect selection, you cannot detect the mutant cells normally because they do not grow. Here I'm going to explain how you can detect a negative selection for a mutant, and that is we have different colonies here on the auger, isolated colonies. You can take a velvet stamp of that where you put velvet on this handle and stamp, and you press the stamp onto the auger. That will put cells on the velvet, and then you can stamp this plate here and that plate here. These plates differ in that this medium contains the amino acid histidine. This media lacks the amino acid histidine. You then look to see where the colonies are growing, and this colony, let me see if I can remember now. This colony here, I think, that one right there, it's stamped right there. You can see that colony in the media containing the amino acid histidine, but it does not grow in this plate here, lacking the amino acid histidine. So we know these cells are mutant, and for these cells to grow, you have to supply the amino acid histidine, because these cells here cannot make the amino acid histidine, so they cannot grow on media lacking that amino acid. And so these cells are a mutant that we've detected by negative selection. Any question about any of this? I actually did this stamping, looking for mutants when I was an undergraduate in college. All right, let's switch gears a little bit and talk about, genetic recombination. Recombination is the exchange of genes between two DNA molecules. Recombination often occurs in crossing over. Crossing over is where two chromosomes get together and pair. The mother's chromosome A gets together with the father's. Actually, it's the same chromosome. So let's make a copy of chromosome A, meaning the first chromosome, the mother, the first chromosome of the father. And you can note that they have different genes. The mother's chromosome has lowercase e, lowercase f, lowercase g, etc. The father has uppercase genes, all on the chromosome. Well, in the child, the mother and the father's chromosomes can pair up like this, and then they can cross over like that, and then they break, and then the mother's chromosome here gets together with the father's chromosome here, meaning this part of the chromosome is the father's part of the chromosome, capital H, and this part of the chromosome is the mother's part of the chromosome. This is crossing over, where two homologous chromosomes, meaning the chromosome from the mother and the chromosome for the father, like the first chromosome of the mother and the first chromosome of the father, come together, cross over, and then break right there. Recombination often is crossing over, but recombination doesn't have to be crossing over. Recombination is a little more general, and wherever two chromosomes exchange genes between them, that is a recombination event. For example, let's say the HI gene of the father were to break off the father and then add to the mother's first chromosome. So the mother's first chromosome has both the lowercase h gene and the lowercase I gene, but also has the father's capital H gene and the capital I gene. That doesn't happen very often, but if it were to happen, that would be a recombination event. It is not a crossing over event. Any question about any of that? Crossing over normally only happens only in the homologous chromosomes, meaning the chromosome of the mother and the chromosome of the father, where the genes are either the same or they're very similar to each other. And they are the same, well, loci. They don't have to be the same alleles, but this gene here, if it's coding for eye color, that would be, come on, mouse, that would be coding for the blue eye gene, and that could be coding for the brown eye gene. All right, any question about recombination? So let's move on to talking about genetic transfer. We talked about vertical gene transfer. This occurs during reproduction, where the genes transfer between generations of cells from one parent cell to two daughter cells, or in the case of reproduction, like human reproduction, where the genes transfer from two parents to one offspring. And that's vertical gene transfer. We can also have horizontal gene transfer, where genes transfer between cells of the same generation. Between a donor cell giving DNA to a recipient cell. So the donor cell gives the DNA and the recipient cell takes the DNA. Horizontal gene transfer can happen in bacteria in 3 different ways: by transformation, conjugation, or transduction. Transformation is where DNA comes out of one cell, the recipient cell, and then you get naked DNA in the environment, and that naked DNA can be taken up by the recipient cell, shown here, and then that lowercase a gene can combine with the chromosome, capital A, and now this cell has new DNA, the lowercase a gene, which was not in the recipient cell. It came from the DNA coming out of the donor cell. And so this cell picks up new DNA, the lowercase a gene, from the donor cell. Oftentimes that donor cell dies, and that's why we got naked DNA out here, which can be picked up by the recipient cell. An example of this would be if lowercase a gene codes for drug resistance and capital A does not, this cell can then pick up the gene for drug resistance by picking up the lowercase a gene. Any question about any of that? Here we're looking at transformation, where the cell takes up some DNA it finds in the environment, gets some indigestion here as the gene is expressed, and then the phenotype of this cell is changed by the DNA that it took in. This phenotype of the cell, that's the phenotype of the cell previously. Any question about that? So that is transformation, where the cell picks up new DNA, which can then change the phenotype of this cell. Such as if this gene here contains antibiotic resistance. Let me stop the show here and talk about the importance of that. All right. Spontaneous mutation can change that gene so that for every gene, one times 10 to the 6 genes. I'll make that a little better in a minute. A superscript that. Oh, crud. Make this bigger. The spontaneous mutation occurs one gene mutated per every 10 to the 6 genes. So we could get, if we're looking at antibiotic resistance, one gene can be mutated per every 10 to the 6 genes. But transformation can increase this frequency one gene mutated per every, make that a little smaller. 10 to the 5th genes. Oop. So that transformation can happen 10 times more frequently in bacteria than mutation. So if we're looking for spontaneous mutation to a drug resistance like penicillin resistance, you're going to have to look at 10 to the 6th cells to get one mutation. But if you had transformation happening, it will happen 10 times more frequently. And let's go ahead and just say conjugation in bacteria, as well as transduction. We'll talk about these in a minute. Each of these happen 10 times more frequently than spontaneous mutation. Any question about any of that? So that's telling you the importance in bacteria of transformation and conjugation and transduction. They're going to happen in a cell 10 times more frequently than spontaneous mutation. All right, let's talk about conjugation. Conjugation can happen in two ways, depending on the cell, if it is a gram-negative cell or a gram-positive cell. In a gram-negative cell, we have conjugation happening through a sex pilus, which is this thing here, which literally skewers, excuse me, this cell here. It comes from that cell, not from that cell. And then the plasmid in this cell can move from this cell into that cell. That's sexual conjugation in a gram-negative cell, but we have sexual conjugation in a gram-positive cell, and that happens through a mating bridge where two cells get together and the cell wall is removed in this region, and then the plasma membrane here fuses to the plasma membrane there, and now the cytoplasm is actually connected between this cell and that cell here. So the cytoplasm from this cell can move there, and the cytoplasm from this cell can move there, and we could have like a plasmid moving out of this cell and then moving into that cell. And that is how sexual conjugation can happen in a gram-positive cell. Any question about that? We won't talk any further about the mating bridge, but we will talk further about conjugation through a sex pilus. You have a cell here that has a chromosome and a chromosome here and a plasmid here. This is the F plasmid. The F plasmid literally codes for the sex pilus, which is not shown well here. So let's go previous. There's the sex pilus here. This cell has the plasmid that codes for this sex pilus being made. The sex pilus, what it does is it finds an F minus cell and then literally skewers that cell. So this one here creates the sex pilus. It goes out, finds an F minus cell, skewers that cell, and then the sex pilus can act as a winch and bring the two cells closer together. They do not need to be this close together because the sex pilus is hollow, and even if the cells are this far apart, the plasmid in this cell can actually move through this hollow sex pilus and then move into that cell. The plasmid, before it moves, it will replicate itself. So this plasmid now has two copies of itself in this cell. One copy will be sent through the sex pilus to the F minus cell. This F minus cell now has the F plasmid in it. The F minus cell has the plasmid to make the sex pilus. So we'll begin construction of the sex pilus. And once construction is finished, this cell can then go out and skewer an F minus cell, replicating its plasmid and then sending it into the F minus cell. So this F minus cell is no longer an F minus cell, it's now an F plus cell. And that's how sexual conjugation can happen in a gram-negative cell through the sex pilus. Any question about any of that? All right, transduction happens in a bacteria by using a virus. Let me briefly talk about a virus. A virus will be a protein, the blue-green color, and then the red-purple being the, let's just call it viral DNA. When the virus binds to its host cell, the virus will inject its DNA into the cell, the viral DNA into the cell. The virus DNA, one of the first genes that will be expressed, it will code for the destruction of the bacterial chromosome. Why? The virus DNA wants to call the shots in this cell, and an easy way to call the shots is to destroy the host cell's chromosome. So one of the first things it does is it creates an enzyme which cuts up the host cell's chromosome. And now the viral DNA is calling the shots in the cell. What the virus will do is cause this cell to replicate more viruses. And so it'll cause the cell to replicate its viral DNA and then replicate the viral protein. The viral protein will get together with the viral DNA. And then the last thing the viral DNA will do will code for the destruction of the bacteria cell, and that will release the new viruses. But when viruses replicate, they make lots of offspring. So if they make a mistake, they're not terribly worried about it. Let me blow this up a little bit if I can. There we go. You can see that this here is normal viral DNA, meaning it has the red-purple color in it. But this one there has a dark purple color in it. The DNA that was put into this virus is not viral DNA. It is DNA of the host chromosome. Now, when this virus binds to this cell, it will insert its DNA into the cell. But what's being inserted into the cell is not viral DNA. It is DNA of this chromosome. And then this DNA can combine with the host chromosome of the cell so that this cell now gets new DNA. And it's the DNA of this donor cell, which has been killed by the virus right there. But my point is, this DNA can be incorporated into the chromosome, and then that DNA can be expressed, giving this cell new genetic information. For example, if this were a piece of DNA which were to contain the antibiotic resistance gene on it, and that were to be got into this virus, and that virus were to inject its DNA into this cell could become resistant to antibiotics by that DNA incorporating into the chromosome of this cell. And that's how transduction occurs in a prokaryote cell. Transduction probably happens all the time in the nose cells, maybe the throat cells of humans. But who cares? Because if my nose cell gets somebody else's DNA, when I die, or my node cell dies, either one, that's going to be the end of that other person's DNA. And that DNA will not be important to the human genome because it was not passed on to the offspring. So transduction, for it to be important in humans, you'd have to have somebody's germline cell pick up DNA from another individual, and then that germline cell would have to go on and create an offspring. That can theoretically happen, but it doesn't happen very often. And I know of no examples where anyone's ever found a case of that. But like I said, in my nose and my throat cell, I probably have transduction happening all the time. But who cares? Because that won't be passed on to the next generation. Any question about any of that? All right, we have 1 minute left. Let's talk briefly about plasmids. Plasmids are a small circular piece of DNA outside the chromosome, why it's called extrachromosomal. It does contain genes, but only a few genes. Like this plasmid here has a handful of genes. The chromosome, on the other hand, has, well, human chromosomes have thousands of genes. Bacterial chromosomes could have tens or even hundreds of thousands of genes on them. So the chromosome has many more genes than a plasmid. A plasmid can replicate independently of the cell's chromosome. It usually does replicate when the chromosome replicates, but it can replicate independently. For example, right here, no, right here, this plasmid replicated independently of the chromosome, making another copy itself, and then sending one copy into this F- cell. So that plasmid was replicating independently of the chromosome. Plasmids are important because they're small pieces of DNA, and bacteria cells are more likely to exchange a plasmid than they are a piece or the entire chromosome. So plasmids are more likely to be exchanged among bacteria cells, and that can increase genetic variation as the cells in the population are exchanging plasmids. And they're more likely to exchange a plasmid than they are DNA on the chromosome. Any question about any of that? Let me state that there are different categories of plasmids. You don't need to know that. All you need to know is there are different categories of plasmids. Any question about plasmids? If not, I'm going to stop here, and I will be in the lab at 6:30 at the normal time. You don't need to show up unless you want to ask a question or you want to listen to questions. Any questions? Whoops. All right, I'll see you.